infertility.
Okay.
Okay. Thank you.
Okay. Merci beaucoup, Medhat, because you know you are French speaking. I would like to thank the organizer for this kind invitation. And I will at least mention what's our policy for genetics of male infertility investigation in our department in France. As you know, that's for male infertility, there is a first line investigation. And we know, especially if you are dealing with an reproductive genetics department and IVF, we know that's 20 to 25% of male, they are coming for genetics and fertility. And the first exam to be done, because it's reimbursed by the state, is the karyotype. That means the chromosome analysis, followed by, based in the results of the spermogram, it's followed by the chromosome Y microdeletion. And if you investigate the potential contribution of chromosome abnormalities, you have a numerical chromosome disorders around 15%. And the main abnormalities is Klinefelter syndrome. But also, please don't ignore the contribution of structural abnormalities. It's around 1%. And this can be Robertsonian or reciprocal translocation. And also, that's a chromosome Y microdeletion is extremely important. Now, if we investigate the karyotype and the chromosome Y microdeletion, and the strategy it will be, we should try to make the correlation between the phenotype and the genotype and TESA success chance. That's clear. We go for this genetics testing. We will make the correlation between genotype and phenotype. And from this, we will make the prediction of the TESA success. This is very important because we should get at least the minimum of information to decide to go to the TESA or not. And here the example is the common examples for chromosome Y microdeletion. This is the azadef gene. You have the sperm phenotype, it's an azoospermia or oligoazoospermia, and the testicular phenotype will come with Sertoli cell only syndromes, with the meiosis blocking or the hyperspermia case. And if you have the spermatogenesis, you have nearly 50% of chance to collect the sperm from the testis. And even if the azadef is deleted, also it's a good marker to get positive chance, nearly 50%. But please be careful, it depends who is doing the biopsy. It's very interesting. This is also should be considered. And now the question is, are you also sending, all the biopsies when you do it to the pathologist or not? Now, we are in the era of genome sequencing. The sequencing technology now is coming affordable. Especially if you, for my colleagues, if some scientists are here, that's a clear. Illumina and Thermo Fisher, they are not the only leaders because they are killing us. Now there is a new possibilities with Oxford Nanopore. That means there is a small sequencer now. The cost is around 5,000 euros. Now, how we can implement the sequencing for diagnostics and prognostics? That's clear. We are interested about the primary infertility or non-obstructive azoospermia or idiopathic. From that, we go to the whole genome sequencing. I won't mention the detail. It can be short read or long read. This is your scientist. From that, we were setting what we say the gene panels in silico. We investigate the data. We go to the bibliography database. We make the correlation between the genotype and the phenotype. And from that, we will assess the chance of TISA, and we will go for TISA, or we wait a bit and try a second assay. This is here now, science is moving, and we are getting a key information when you want to make the predictive correlation between the genotype and the phenotype. I mean, especially when the sperm donation is not allowed. That's clear now, based in the, when you go to the exomes and you, thanks to the bioinformatics. Also what I want to say, even if you don't have the bioinformatics, if you invest for small sequencers, you can send the raw data to some companies if your government is allowed you to send the information abroad. For example, in France, the government not allowed us to send the raw data from our sequencer to China. This is also problematic. But even in Egypt, even if you can run the test in your lab, because for me, we should stop to talk about sperm lab. We should talk about molecular andrology laboratory. That, I mean, doing spermogram and spermiocytogram like 40 years is not pertinent anymore. That means the real-time PCR should enter to our andrology lab, and even the immunosequencing will come. From this, you can see the picture where you should go, and from that, when it's green, it's green light to go to the sperm retrieval, and you have good chance to find the sperm. But now it depends also who is doing the TISA. When it's red, I think it's not, I mean, promising situation. Now, if we go to the exome sequencing and the bioinformatic data analysis, I don't want to disturb you a lot about what we say, the CNV and the VU. As you know, that's 4% of our genome is in continued dynamic mutation. And sometimes also this is the complexity of the interpretation of the data. Because we have some variant, we don't know if they are pathogenic or probably pathogenic, or they are of the non-known significance. This is also the power of the technology, because in the end of the day, it's extremely different sometimes to send you the final report, because we are not able to conclude from the data coming in our hand. This is also, you as a clinician, you should accept times to time this situation coming from us. And here you can see, at least in my team with who I am collaborating, because, and we identify in each steps of the meiosis, of the spermatogenesis, the key gene that you can consider it, and from that, based, because the best education, training and education for the clinician is his own office, because the profile of the patient can be different. From that, you can identify what we say, the very well specific panels, and from that, you can go very fast for the turnaround time of for your patient, what we say the mini sequencing technology. And for examples, with my groups, we select only the green and the red gene to be considered. From that, the cost will be less expensive, and also the turnaround also. But here, when we want, our panel is containing 151 gene for the exome sequencing, or we can say the mini exome sequencing. And we identify from this 151 gene, 12 gene subjected to variants. And this should be considered and it was in concordance, pronostic concordance about 100% for the TISA, for negative TISA. This is an example. We have one patient, non-obstructive azoospermia. This is our patient number 20. The cytogenetics testing, we went for the karyotype, the molecular AZF microdeletion. This is the first line entering for the. After we went to the whole exome sequencing, and we identify a variation in 142 non-candidated gene from the 151. After we identify a rare variant, this is give us a low chance, and TISA was counterindicated. Because with this variant of MeO-B gene, it's an pathogenic mutation in male, and this is the cause of meiotic arrest. From that, probably you stop, managing the patient if you should go to the sperm donation or cancel the file. And when our colleagues, Zinedine El-Kirraf, make the knockout models, because within the lab research, this meiotic specific for the OB fold, it's causing meiotic arrest in male and female mice. Also, this is also if you are introducing to the sequencing technology. This is the problem of non-obstructive azoospermia and predisposition to the cancer genes. In 2014, with my PhD student, D.B. Mongeon, now she's directing two of my embryology lab, we identify that in male infertility, during the meiosis, some gene involved in cancers can be mutated. Now it's confirmed by sequencing because some genes in male infertility, mainly the genes involved in DNA repair and the cellular homeostasis. You can identify via the NGS some patients, even if they are not fully infertile because they are positive for TISA or for ICSI, there is a risk if you have a premutation of some genes involved in cancers, and this can get a global negative consequence for the patient, and this is you should inform the family about the risk of the comorbidity and the cancer predisposition risk, because you have the information, but you should inform the family, because if one day something's happened, because you said, why you didn't inform me about this risk? And now we are facing some difficulties. Now you tell me what I should do for this patient. That's clear. We should be very careful about the transmission, prevention of the transmission. If you decide to go to ICSI with this patient, please don't forget to offer to them the PGD or the prenatal diagnosis of this risk. This is another example. The same things, non-obstructive. We want, in the end, TISA was not recommended because we identify a variant for Fanconi anemia. Fanconi anemia is a DNA damage to the sponsored genes, and from that we identify a risk for a mutation for breast cancers. Again, this is pathogenic mutation in male, and this is causing the testicular degenerescence syndromes, and in the knockout show the infertility also. Now if you go to the macrospermia, this is also a simple test can be identified. This is the ORCA gene. This is mainly the common mutation in North African patient because the ORCA gene is a male North African mutation. And in this case, the problems of the ORCA gene is an communal, and from that we investigate all 150 56 patients analyzed with Sanger methods, we identify 50%, 58% of the patient with the mutation. But when we went to the exome sequencing, we identify other 6 new genes involved in the macrocephalicans. But please, for your patient, don't investigate only the ORCA gene. There is other 6 genes that needed to be considered. Now, from that, there is an algorithm now coming. If the sample is addressed for genetics diagnosis of severe sperm defect with large head spermatozoa, we know if we have more than 20% of large head and more 20% of multiflagella spermatozoa, I think to go faster, we go to the Sanger technology by investigating ORCA-C exons 3 and the other 6 gene what I mentioned. If it's positive, we go to the reporting, but if it's negative, we should go to the whole exome sequencing. For the globulospermia, you know that is not communal, but it's existing. You know the problem of the globospermia is the mutation of one gene causing the absence of the PLC zeta. Now, in my laboratory now, the PLC zeta by immunohistochemistry is a routine test, alas, because you know many colleagues are criticizing the DNA fragmentation or the chromatin denaturation, but we are using as a routine test. In globospermia, 78% of the patient having a homozygous deletion of the gene DPY19L2. But you should know, this is what we say the normosperm infertile. We have some patient, the spermogram and the spermocytogram is normal, but when you do ICSI or IUI or IVF, there is missing of fertilization and activation. This is the PLC zeta history. You know that the PLC zeta is the key factor of oocytes activation after ICSI. Now, this is our algorithm now to do it. If we have the mutation of the PLC zeta, it's a clear indication for oocytes artificial activation. And also, if you have the mutation of the gene, you have a risk of the retention of the histones and the DNA fragmentation, and this is a bad prognosis for ICSI. For the acephalic spermatozoa, now there is one gene identified responsible about that, is the SUN5 protein or the product of the gene. But the data coming from the literature in North African patients showing this is the common variant for this acephalic syndrome, and the data published by the Chinese groups showing that the SUN5 mutation is not affecting the IVF or the ICSI outcome. For the MMF, you know, this is the problem of dyskinesia, and this is also not common. This is our algorithm, at least in my groups. When we have the macrospermia, we will investigate the ORCA-C gene. In the globospermia, we are mainly concentrating in DPY19L2, and in the acephalic syndromes, we are investigating the SUN5 gene. Ladies and gentlemen, today you can investigate those genes by simple real-time PCR. You know, after the COVID, the majority of the laboratories, they have hundreds of PCR machines in the corner. Take one, and even the microdeletion of the chromosome Y now can be done by real-time PCR. And later on, if it's needed, we have a specific panel to help you to manage your patient suffering with abnormal sperm. And thank you very much.
Thank you very much, Moncef.